Good afternoon to all of our Caltech alumni and friends of the Caltech community. My name is Helen Kim, and I'm pleased to welcome you to today's webinar, Deceiving the Superorganism, Infiltration of Ant Societies by Stealth Beetles. Today's presentation is hosted by Dr. Joe Parker. Joe's presentation was very popular at our 81st annual seminar day that was held in May on campus. We are pleased to be able to bring his talk to a wider audience today. Joe Parker is an assistant professor in the Division of Biology and Biological Engineering. Prior to arriving at Caltech, he received a Bachelor of Science in Zoology from Imperial College London, did his graduate work at the University of Cambridge MRC Laboratory of Molecular Biology, and was a Wellcome Trust postdoctoral fellow at Columbia University and a research associate at the American Museum of Natural History. Joe is an entomologist whose work addresses a fundamental question in biology. How predictable is evolution, and to what extent is evolutionary change predetermined by ancestral conditions? He established a unique model system to address this question: rove beetles that live symbiotically inside colonies of ants and termites. Such species embody evolution in the extreme, with dramatic behavioral, anatomical, and chemical adaptations for life as social parasites. Joe found that these adaptations have evolved convergently many times, providing a paradigm for understanding how complex changes in organismal biology can arise predictably over evolutionary time. Using his training in fruit fly genetics, Joe's lab is now using rove beetles to dissect the molecular and neurobiological changes that underlie the convergent evolution of social behavior and interspecies interactions in this remarkable group of insects. During the last 10 minutes of the presentation today, Dr. Parker will answer questions from the audience. Please feel free to write in your questions at any time during the webinar using the questions feature within the platform. And without further ado, I'll turn it over to Dr. Parker.
Thank you so much, Helen, for that nice introduction. And thank you all for tuning in to see this webinar. It was a ton of fun to be able to do this on seminar day, and it's great to have the opportunity to do it again for all of you who may have missed it on the day itself. So without further ado, I'm going to get going and give you a talk about something which has really been a lifelong passion for me, which is the insect world and a specific facet of it, which has become the main program of my research here at Caltech. So I put up this picture here of these ants. You may be able to see, if you stare at it for long enough, that some of these ants look a tiny bit different to the other ants in this image. So if you can see what I'm doing with my mouse here, you see this ant here, its abdomen slightly yellow, its anterior body is a slightly different shape to the other insects in this picture. That's because this ant here isn't actually an ant at all. It's in fact a beetle that lives among these ants and is accepted by them as a nestmate. And that sounds really harmonious, except for the fact that this beetle feeds on the ant babies as this really fiendishly deceptive colony parasite. And I'm going to discuss with you my work today on these beetles and this way of life, and how it provides a really interesting window into a pervasive phenomenon in the natural world, which are the symbiotic interactions that have evolved between different animal species. Now, probably every animal out there is engaged to some extent in a symbiotic relationship with another animal species. These range from mutualistic relationships, where both partners contribute to the symbiosis and it's a kind of shared benefit, to parasitic relationships, which are much more common, where the relationship is very one-sided and one organism profits at the expense of another one. You can think of cleaning mutualisms, for example, cleaner fish, and brood parasitism in cuckoos and cowbirds as examples of these extremes of symbiosis. And symbiosis is just an umbrella term that means living together. So when two organisms live together, whatever the nature of the relationship, we can refer to it as a symbiosis. Of course, these kinds of symbioses extend into the human realm, and the parasites that we harbor and animals we've domesticated. If you think about what a dog is, a dog is really just a symbiotic wolf that lives with a human host, and it's debatable whether that's a really mutualistic or parasitic relationship. It probably depends on how well behaved your dog is. And so it's a really basic question in biology to ask why organisms become symbiotic. Why do they start to engage in interspecies relationships? And then also, what are the underlying molecular, genetic, neurobiological changes that happen when species engage in a symbiotic lifestyle, when they leave an ancestral free living existence and start to become obligately dependent on another species of organism? And this is really the question that my laboratory is interested in. And the group of organisms who study this question in are really the world champions at evolving symbiotic relationships. These are the rove beetles, the family Staphylinidae. If you've never heard of rove beetles, well, let's change that. Rove beetles are actually the largest family of organisms within the entire animal kingdom. There's 63,000 described species of these beetles. There's more of these rove beetles than there are vertebrates. So if you add up all of your fish, your birds, your amphibians, your reptiles, your mammals, there's still more of these rove beetle species. And that 63,000 known species is maybe 5 to 10% of the total number of rove beetle species out there. So if you go to the woods in Southern California and look around in the leaf litter and dirt, you can find new species of rove beetle that people have never seen before. They're what's called a hyperdiverse animal group. Now, like I mentioned, these things largely live in the dirt. They're small beetles, usually under a centimeter long, and they live in leaf litter and soil habitats and live a kind of predatory existence where they feed on other microarthropods down in the dirt. And you can see they don't look like your typical beetles. Rove beetles are characterized by having this elongate exposed abdomen. They have short wing cases. All beetles have wing cases. That's a diagnostic feature of beetles that usually cover the whole abdomen. But in rove beetles, they leave the abdominal segments exposed, and these segments are flexible and telescoping, and it enables these beetles to snake through leaf litter and soil habitats where they chase down arthropod prey. So there's this just gigantic group of really tiny predatory beetles that live in the dirt, and by and large the biologies of these beetles are quite boring. They just, feed on other insects, but repeatedly during their evolution they've gained the capacity to do something amazing. Whereas most insects that would wander into an ant colony like this beetle here would be recognized and attacked and dismembered and turned into food, ants have this really sophisticated mechanism to recognize intruders. On their bodies of the worker ants, they have cuticular hydrocarbons that they use to recognize nest mates, and intruder organisms which don't match that cuticular hydrocarbon code are instantly recognized, and the ants are programmed to attack anything that doesn't match that cuticular hydrocarbon code. So this beetle here is instantly detected by these ants, and they start to attack it. Now, multiple independent evolutionary lineages of rove beetles have evolved to bypass this mechanism of nestmate recognition. So if we compare that beetle to this one here, look what happens. This beetle here, far from being attacked by these ants, is actually being fed mouth to mouth by this ant and being treated as if it were just another one of the workers. The ants will treat this beetle like royalty, allow it to kind of wander through the nest, feed it mouth to mouth like this, and it lives the life of Riley as this kind of deceptive colony parasite. How is the beetle able to do this? Well, it's able to chemically deceive these ants. Ants live in a chemical world, and so different groups of invertebrates have evolved to chemically trick ants to infiltrate their colonies. In the case of this beetle, if you look on its back, it's got these yellow brushes of hairs, okay, and you'll see they're called trichomes, and they are kind of biological candle wicks that secrete compounds from glandular structures at this part of the abdomen. You'll see ants wander up to this part of the beetle and start to lick it really vigorously. They're really attracted to whatever's being secreted from these trichome structures, and it exerts a kind of chemical mind control over the ants. It suppresses their aggression towards the intruder beetle and causes them to adopt the beetle into the colony. So it assimilates into the social fabric of the ant colony. And the ants will even pick the beetles up by this part of the body, carry them around the nest, dump them in the brood galleries, where this beetle will pierce open the ant eggs and suck out the insides. Again, this is an amazing mechanism of chemical deception of ants that enables these beetles to assimilate into the nest. Now, insects that have evolved the capacity to live inside ant colonies are called myrmecophiles, ant lovers. And across the tree of life of insects, you see this lifestyle evolve sporadically. So there are examples of myrmecophilous crickets, which are integrated into ant colonies. There are even there's a whole family of butterfly caterpillars, Lycaenidae, which again are socially integrated inside ant nests and wholly dependent on them during their larval life. Whenever you see this kind of lifestyle evolve, myrmecophily, it's really an amazing example of just how intimate interspecies interactions between animals can evolve to be. They're really behaviorally intricate associations that involve all of these really amazing behavioral and chemical adaptations, where these insects have sacrificed the capacity to live outside ant colonies, are totally dependent on their ant hosts, and have evolved all of these ways to socially assimilate into nests. Now, by far the largest number of independent evolutionary instances of myrmecophily are found within the beetles, the order Coleoptera. There's 400,000 species of beetle. 1 in every 4 living things is a beetle. And when beetles evolve myrmecophily, often they show some of the most dramatic adaptations to this way of life. And this is evidence in the crazy anatomies that these myrmecophile beetles show. And I'll just give you a couple of examples to demonstrate what happens when beetles evolve into myrmecophiles by comparing a couple of really dramatic myrmecophiles to their free-living close relatives that probably embody the ancestral free-living condition that they evolved from. So, for example, this is a ground beetle, a family Carabidae, a thing called Harpalus. It's a free-living generalist predator. You may have seen similar beetles like this wandering around, just feeding on things like earthworms and stuff like this. Now, this is a version of a ground beetle which has evolved to live inside an ant colony. So this is a myrmecophile version of the beetle on the left. It's called a porcine carabid, Platyropalopsis, and you can see it's the most amazing thing. These Mickey Mouse ears up here, these are actually the antennae of these beetles, which are massively expanded. All of the segments have fused together into this gigantic plate, and it's decorated with tiny glandular structures that are secreting compounds that again are behaviorally manipulating ants. So this beetle will wander up to an ant colony. Ants will lick whatever's being secreted from these disc-like antennae, and it causes the beetle to be adopted into the nest, where again it does things like feed on the brood as this kind of colony parasite. So this is really dramatic, extreme evolution, because when you evolve to live inside an ant society, selection pressures change, and this is manifest in really dramatic changes in the anatomy and behavior of these insects. I'll give you another example. This is a rove beetle, very much like the ones I was just showing you earlier. Free-living generalist predator, again, very common. This is a species of rove beetle that has evolved to live inside army ant colonies. And you can see it really doesn't look anything like the beetle on the left. All of its dorsal anatomy is essentially evolved into this gigantic shield-like structure. So this beetle is invested heavily in physical defense. It's got this huge carapace that covers it from the top. If you were to flip it round, it would look kind of like a typical beetle. It's got 6 legs, antennae, eyes. It's not too different underneath, but the whole dorsal surface of it has been transformed into this big shield-like structure, again, in response to the hostile, aggressive nature of ant society. So you can see these kinds of 2 different strategies here. There's one of social trickery and chemical deception of the ants so that they accept you. That's one way you can get your foot in the door of an ant colony. The other way is just to really invest strongly in physical protection, so that even though the ants recognize you, they can't actually kill you. Okay, now, one of the most dramatic manifestations of this way of life, this myrmecophilous existence, is found in rove beetles that live inside colonies of army ants. Now, army ants are these tropically dominant ants that form huge colonies, sometimes millions of workers large, that lack permanent nest sites. And so they lead a kind of nomadic existence, emigrating across the tropical forest floor, feeding on other arthropods, and they're highly aggressive colonies of ants. You sometimes get these major workers with these giant mandibles like this, which are really, on guard the whole time. So they're very difficult ants to kind of work with. And you can imagine for most insects in the tropical rainforest, encountering an army ant colony just spells doom. You get swamped and eaten, turned into food. So this is what most army ants do, is they hunt down other insects and feed on them. Now, if you happen across an army ant trail as you're walking through the tropical forest and watch it for long enough, you might notice that maybe 1 in every 1,000 or so of these ants wandering past will in fact be a rove beetle that looks like this. It has amazing ant-like morphology, this so-called myrmecoid ant mimicking body plan. And it's not just the case that these rove beetles are anatomically mimicking the ants, they're also accepted into these really hostile, aggressive ant colonies as if they were nestmates. Again, they are these socially symbiotic, socially parasitic organisms which have assimilated into these aggressive army ant societies and are profiting from the resources that the army ants have. They may feed on the brood, or they may feed on the arthropod food items that the army ants have harvested. Their biology is completely synchronized with the host colony, so they emigrate with them each day. They may hang out with them in temporary nesting bivouacs inside holes in the tree. They are, to a large extent, behaving just like the ants themselves, except they are beetles and they're not contributing anything beneficial to the colony. And there are several dozen different genera of these ants, all of which have this dramatic myrmecoid morphology, and they're found with army ants throughout the world's tropics. So you can see it's the kind of evolution of this really complex symbiotic syndrome whereby these ancestral rove beetles like this evolved into these ant mimicking ones that have all of these social behaviors. So they live with the ants, they groom the ants groom them, they're accepted like nestmates into the nest, and it all evolved from these really simple kind of predatory beetles that look like this. And it's been controversial how many times beetles like this have made the leap into these myrmecoid symbiotic beetles, whether it's happened once, there's been a single origin of these beetles and they've co-diversified with army ants throughout the world's tropics, or whether beetles like this have made this leap multiple times convergently, which would be really extraordinary evolution of this really complex symbiosis. So to shed light on this question of the evolutionary origin of these amazing symbiotic beetles, together with Munetoshi Maruyama from Kyushu University, we collected these myrmecoid rove beetles from army ant colonies throughout the world's tropics, so Southeast Asia, the Afrotropics, and the Neotropics in South America, and sequenced their DNA to resolve their evolutionary relationships using molecular data, really to shed light on how many times these beetles had evolved, whether it was just a single origin or whether it evolved multiple times. And this is what we found. It was clear that this is a trait, a symbiotic lifestyle that had evolved multiple times, at least a dozen times independently across the rove beetle tree of life. So you can see this is the phylogeny, the evolutionary tree that we built using DNA sequences. All of these black lineages here, they are rove beetles that have this typical rove beetle morphology, most of which don't live with ants. So this is kind of free living backbone of the tree, which is seen here in these black lineages. But repeatedly, all of these little orange groups, these independent little lineages or clades, have branched off this tree. Each one of these is independent of the other ones here, and each time giving rise to these really dramatic ant mimicking beetles that you see along the top here. So you get the repeated or convergent evolution of these beetles right across the tree, and it's really rampant, frequent evolution of this kind of symbiosis. So just to clarify what I mean by convergent evolution means the repeated evolution of similar traits in response to similar selection pressures. So famous examples of convergent evolution are the evolution of fins in whales, penguins, fish, and ichthyosaurs, and the evolution of wings in animals that can fly, in birds, butterflies, pterosaurs, and bats. Now, these are really, classical examples of convergent evolution. What's so amazing about this system here is its convergent evolution of an entire symbiotic syndrome, where the entire lifestyle and anatomy and behavior of these insects is all evolving in the same direction. It's not just a single trait that's evolving, it's the entire life history of the organism. So it turns out each one of these independent evolutionary instances of this symbiosis is associated with a different army ant genus. Okay, so what this means is every time a beetle like this has evolved to ecologically associate with army ants, its morphology and aspects of its behavior have walked down a really predictable evolutionary pathway to produce this convergent evolution of this complex symbiosis. It's this really dramatic example of convergent evolution that's without real close precedent in biology. Many of these lineages are separated by as much as 100 million years. So what that means is that it's a deep time system of convergent evolution. It's a highly predictable, deterministic pattern that we see happening right across this vast group of animals. And it enables you to ask how repeatable evolution is at the molecular, genetic, neurobiological levels to produce this really elaborate phenotype in each instance. Now, just to kind of emphasize how strikingly convergent these lineages are, I've given some examples here of these beetles which are unrelated to each other, but have each like evolved into really similar morphologies. So, for example, this is a species from South America, a Cytocryptus, that lives with this, evolved to live with this Noma myrmex army ant here. Completely independently, this beetle here, Weissflogia, which lives with Anictus army ants in the Southeast Asian tropics. Now, each of these guys is not really related to each other. They each evolved from ancestral rove beetles that looked similar to these two here. So the degree of convergent evolution is stunning. Here's another couple of examples. Again, this is a New World tropical species, genus Diploeceton, and this is an Old World tropical genus, Giraffinictus, each having evolved to live with different army ants, each evolving from ancestral beetles that must have looked quite similar to these two here. So convergent evolution in the system is really, really amazing at the morphological level, but I want to emphasize that it's behavioral convergence too. Each one of these ant-like symbiotic beetles has evolved similar social behaviors. So if you see one of these beetles interacting with ants, they're treated like royalty. So the ant here is grooming the beetle. You know, it's like cleaning it, it'll give its antenna a good old lick. They're really perfectly accepted, never attacked inside these colonies, and all carrying out these social behaviors, these grooming behaviors, things like feeding, being fed by the ants, essentially behaving like they are worker ants. So it's anatomical and behavioral convergent evolution in the extreme. And I just want to show what happens when I try and socially integrate inside an army ant colony. This is what happens. This shows you quite how aggressive these ants are. It's just by putting my hand into a colony of them with a gardening glove, you get completely annihilated. And that just shows you how dramatic it is that these beetles have been able to infiltrate these colonies where they live really having a good life. So in this system, this rove beetle system, we see beetles evolving from a free living condition to a really complex symbiotic condition where they can no longer live outside these ant colonies. We find it hasn't just happened once, it's happened at least a dozen times, maybe 100 times independently. So if you're interested in this question of how relationships between different species of organism can evolve and how they're controlled molecularly and genetically, you can do no better than study rove beetles, because they are head and shoulders above any other group of organism having this capacity, this predisposition to evolve from a free living existence into a symbiotic one. And it's not just ant colonies that they've infiltrated, it's termite colonies too. So they've been equally successful at infiltrating the social structure of termite nests. Now, termites obviously look a little bit different to ants. And when these beetles have evolved into so-called termitophiles, termite lovers, they've been able to mimic the termites again. So this organism here is in fact one of these rove beetles that lives inside a termite colony. This is the host termite here. This is the beetle walking behind it, and you can see it's got this really remarkable anatomy. This beetle, in fact, emerges from the pupal case looking quite similar to this one here, but begins feeding and feeding and feeding and feeding, and its a fat body inside it expands and expands, and it swells up into this kind of grossly overgrown, obese organism, which now starts to resemble the shape of its host termite. So it's this really remarkable phenomenon, this kind of transformation that happens in the adult stage where these beetles grow to mimic their termite hosts. And we know that this termite mimicry has also evolved convergently multiple times within rove beetles. So these are 4 examples of these convergent termite mimics, each of which independently has solved the problem of living in termite nests by converging on this termite-like morphology. You can see this one here, the beetles being fed mouth to mouth by the termite. So again, these are socially integrated inside the colonies of these termites. And we think that in both the case of the ant mimicry and the termite mimicry, the reason that they resemble their host is not that they look like their host, because often the termites and the ants are practically blind or have very limited vision. But what's more important is that these beetles feel like their host. So they're mimicking tactile signals that the ants and termites may be using to recognize each other. So to really gain acceptance inside ant colonies, not only army ant colonies and termite colonies, not only do you have to smell like your host, you may also have to feel like your host too. So we wondered quite how far back during evolution these beetles have been, had the capacity to dupe their social insect hosts, because they seem to be so predisposed to do it and have done it so many times during evolution. Maybe we could look into the fossil record and find evidence for this social insect symbiosis. Now, the earliest evidence of social behavior in both ants and termites is a 100-million-year-old Cretaceous Burmese amber from Myanmar. It's in this deposit, mid-Cretaceous deposit, that you can see evidence of division of labor, of caste differentiation, of collective foraging of these social insects. And it's really right at the dawn of eusociality, at the dawn of complex social behavior in both of these different groups of social insects. And lo and behold, in the same amber deposit, we were able to find examples of these rove beetles, which belong to modern groups that live symbiotically inside ant and termite colonies. So this is the fossil symbiotic beetle we found, Mesosymbion, the Mesozoic symbion. It belongs to a group that has this kind of horseshoe crab-shaped morphology, where this investment in this protective morphology, where the thorax overgrows the head, and the whole beetle kind of looks like a horseshoe crab, or like the beetle I showed you earlier, a trilobite-like morphology. So what the discovery of this beetle means is that right at the dawn of social behavior in both these groups of insects, these beetles had turned up at the party and were already exploiting them as a niche to inhabit. So there's clearly something special about these beetles which has enabled them to become symbiotic many, many times over, and predisposed them right at the advent of the colony formation in social insects to be able to break and enter and exploit colonies as a resource. So what is it about these beetles, these rove beetles, that enable them to make this leap from free living to symbiotic? There are 400,000 described species of beetle. So it's this huge chunk of life on Earth, and rove beetles are just 15% of that. Okay, so across the rest of the beetle tree of life, you don't see organisms with this capacity to make this transition from free living to symbiotic. There's something very special about rove beetles specifically. So here's what I think it is. As I mentioned at the beginning of my talk, rove beetles differ to most other beetles by their possession of these short elytra, these short wing cases that leave the abdominal segments exposed. And this served rove beetles really well for a predatory lifestyle in the litter and soil. It enabled them to snake through these habitats, chasing down other arthropods. But what it also meant was they'd sacrifice the physical protection on the abdomen that most other beetles have. It gave them this kind of Achilles heel, this soft part of the body that left them vulnerable to attack. And they responded to this by, rather than investing in physical protection, in investing in chemical defense instead. So at the tip of the rove beetle abdomen, if you break this segment open, you'll see this reservoir of cells here that contain this yellow substance. This yellow stuff is benzoquinones, and these are topical irritants that enable the beetle to protect itself chemically very effectively. So, for example, if one of these beetles encounters an ant, like this one here, this is one of the rove beetles that we keep in the lab, it can loop its abdomen over, hit the ant in the face with benzoquinones, and run away. And the evolution of this really efficient chemical defense mechanism, we think, has been key to unlocking social insect colonies as a resource. It enables these predatory beetles to wander inside, feed on the resources available inside colonies, things like the brood or the food items that the ants have harvested, chemically defending themselves if they experience attack and running away. And so right across the tree of life of these beetles, they're equipped with this kind of facultative capacity to break and enter social insect colonies. And we think from this incipient kind of starting condition for the evolution of symbiosis, many independent lineages have decided to specialize on ant colonies as a resource. Ant colonies, if you can keep the ants at bay, are predator-free zones. They're full of resources and they're climatically stable. So the selective advantage to being able to maintain a presence inside those nests and profit from those resources is very big. And so there's been repeated selection on these beetles to evolve ways to keep the ants at bay in much more effective ways. And one way they do this is by changing the chemistry that they're using to interact with ants, from compounds that are just the topical irritants, these benzoquinones, which are just, defensive chemicals, to chemicals which can now manipulate ant behavior. And this is a perfect example of this. This beetle here is a rove beetle, Lomacusa. Ants will approach Lomacusa aggressively. It'll loop its abdomen round, just like all rove beetles do when facing a would-be predator. But instead of producing something nasty from its abdominal gland, it produces what we call an appeasement compound that ants find very attractive. They'll feed on this compound and it'll instruct the ant, it'll block any kind of aggression from the ant and instruct the ant to feed the beetle mouth to mouth. So you can see this globule of food being exchanged from ant to beetle here after the ant is fed from a globule of secretion from the beetle's abdomen. After this, the ant will even pick the beetle up, carry it into the colony and deposit it in the brood galleries, so adopt it into the nest. Once it's in the brood galleries, Lomacusa will lay its eggs, and when the larvae of Lomacusa hatch out, they secrete a chemical that tells the ants to feed the beetle larvae preferentially over the ant's own larvae. So this is an amazing mechanism of chemical integration and brood parasitism that these beetles have been able to evolve by having these abdominal glands and this capacity to secrete chemicals which have these profound behavior manipulating effects. So we can see in this system of a convergent system whereby many of these lineages have become symbiotic, there's been really the two key changes that have happened. There's changes in chemistry, which enable these beetles to manipulate ant behavior and gain acceptance inside nests. And there's changes in the behavior of the beetle too, from an ancestral fear-like behavior where they're secreting benzoquinones and just running away, to one of true social engagement, where the beetles are now interacting with ants as if they were the ant's own nestmates. So through changes in chemistry and behavior, there's been the repeated evolution of social symbiosis in this system. And what my lab is interested in studying are the underlying mechanistic changes that mediate this transition from free living to symbiotic by dissecting the genetic and neurobiological basis of these changes in chemistry and behavior. And one way we try and do this is by taking a kind of model organism approach. Now, we can't study symbioses in Drosophila. Drosophila are completely free living. Fruit flies are completely free living. we have to study this phenomenon in these beetles themselves. And so what my lab has been doing is transforming some of these beetles into genetic model systems that we can use the same kinds of tools that we use in fruit flies to dissect the genetic basis, the genetic changes that have happened as these beetles have become symbiotic. So a few years ago, when I was a postdoctoral fellow at Columbia University in New York, I was looking for a species of rove beetle that I could maybe culture in the lab and use as my rove beetle version of Drosophila. And I found that certain biological control companies in North America were actually selling a species of rove beetle by the thousand that you could purchase and dump into your greenhouses as a biological control agent. This species here, it's called Dolotia coriaria. And it's a biological control agent of things like fungus gnats and thrips. So you can buy it in these tubs, litter it into your greenhouses, and it takes control of your pests. And I thought, this is really amazing. I bet I can get hold of this beetle and it'll grow in the lab. If these, evergreen growers supply are able to grow tons of it, I bet I can keep it in the lab, and I bet it reproduces fast. And lo and behold, it did. It's very much, the geneticist's ideal model system. It reproduces really quickly, and you can do actual genetics in these species. And if you look at where it sits in this evolutionary tree of beetles, rove beetles, it sits here. So it's firmly embedded within this group of rove beetles that are predisposed to evolve into symbionts. So what Dolotia coriaria really embodies is the free living starting conditions for the evolution of symbiosis in rove beetles. And so that's what we do in the lab. We treat this beetle as a kind of proto-symbiotic species to understand how changes in its brain and in its glandular chemistry have enabled it, or beetles like it, to evolve into symbionts. And so we're able to grow these beetles up en masse, make millions of them, and do genetic experiments in them. And of course, the structures that we're really interested in are the brain of the beetle and the defensive gland. And we study these structures and how they work and control behavior and chemistry in this species, really as a kind of foundation to explore how these phenomena, chemistry and behavior, have changed in related symbiotic species. And so the symbiotic species we've begun to work with since I moved to Caltech last year. And we found that there are many amazing examples of these beetles that live just on our doorstep up the road in the Angeles National Forest. So Caltech's down here in Pasadena. If you take a 15-minute car ride up to Cheney Canyon just by JPL, you'll find that the dominant ant species in the foothills of the San Gabriel Mountains is this ant Limatopum occidentale, the velvety tree ant. It's everywhere. It's underfoot. It's walking up trees. It's controlling the whole environment in the foothills, wherever you find permanent sources of water, so around rivers and streams. And these ants are extremely aggressive and form huge colonies, maybe hundreds of thousands of workers that hollow out the insides of trees and make their nests. And when we dig into these nests, what we find are some of these rove beetles. So this is the velvety tree ant here, and this is a beetle called Platyusa, which is essentially a version of the free living species that we keep in the lab, but which now lives inside colonies of the velvety tree ant. Now, like I mentioned, velvety tree ant is a very aggressive species, but Platyusa can handle it because it has a trick in its tail. If it is detected by an ant, it just loops its abdomen round and secretes one of these appeasement compounds to the ant, which instantly pacifies it, and ant and beetle go about their days quite happily. So you only find this beetle inside colonies of these ants, and what it's doing is chemically duping the ants almost constantly to keep them at bay. It also mimics the host ant cuticular hydrocarbon profile approximately, not perfectly, but enough to kind of go undetected if the ant's antennae brush up against it. So what we're interested in is how the chemical and behavioral evolution has enabled a beetle like this to now live permanently inside ant colonies. So if we compare the behavior of a free-living beetle, we get this spraying of the benzoquinones and this running away, this kind of classical defense response. We compare that to Platyusa. It's using this appeasement gland that ants find really attractive to keep the ants at bay. And you can see there's no fear of this beetle. it's actually attracted to these ants, and the two organisms hang around with each other without any hostility at all, with this kind of appeasement behavior that this beetle is able to do. So in the system, so just to summarize the behavioral changes that have happened in this system, if you put Dolotia, the free living species, with an ant, you get this primitive defense response where the benzoquinones are secreted at the ant and the beetle runs away. If you put Platyusa with its host ant, it no longer is producing benzoquinones from its gland towards the host ant. It's producing this appeasement compound that the ant finds very attractive, and you get this interspecies social behavior, albeit transient one, but the two organisms hanging around each other with any hostility. But there's a twist to this system that we find very interesting and offers a really basic insight into the evolution of this kind of social symbiosis, which is if you take Platyusa, the symbiotic beetle, and put it with a non-host ant, so an ant that it doesn't live with, you're back to square one. It now uses its primitive defensive gland against the ant. So you can see in this system here that there's been this host ant dependent suppression of this primitive fear response. Somehow, and we think it's smelling like the correct host ant, it suppresses the beetle's urge to use its primitive defense gland, instead to use the appeasement gland, and it leads to the evolution of social engagement. So what we're really interested in is now delving into the brains of these insects to look at their neural circuitry, to understand how a beetle like this can overcome its innate fear of the ant, instead using its appeasement gland instead of running away, leading to the kind of social engagement of the two organisms. And we're able to reproduce these behaviors in the laboratory in beetles that are tethered on walking balls. So this is a rove beetle here, like pushing this ball around, attached to a little pin with glue on its back. And it's got this flexible abdomen. So if you annoy the beetle, it bends its abdomen around and deploys its gland. And so what we're interested in doing now is using genetically encoded calcium sensors to look at neural activity in the brains of these beetles as they're doing these behaviors. As we introduce ants into these arenas, so the beetles can detect their ants, to explore how these different circuits are activating in response to the correct host ant or the non-host ant, to see which novel pieces of neural circuitry have enabled this beetle to suppress its primitive fear-like response, leading to the social engagement in the system. So just to kind of summarize, we think in the system, every time these rove beetles have evolved into symbionts, it's the formation of this kind of basic social circuit, whereby smelling the correct host ant odor suppresses this primitive fear-like behavior, so they do not use their defensive gland. And in return, the beetle is able to pacify this innate aggression of ants towards non-nestmate organisms by using these appeasement compounds. And of course, they're really interested in what these compounds are, because they have these such profound behavioral effects on ants. And so maybe they, as behavioral neuromodulatory compounds, they're really fascinating to us. So one of the projects in my lab is trying to extract these compounds to find out what they are and see how they can exert such profound effects on the brains of ants that feed on them. Now, in the same colonies of these velvety tree ants that we find just north of the Caltech campus, we find another species of beetle wandering around. You can see it in these in amongst all of the chaos. You'll see these beetles wandering around. They're a bit smaller than Platyusa. You can see I'm just pointing to it with my cursor here. This beetle is really, really remarkable. It's called it back into the lab and put a Skeptobius and an ant in the same container. You see this kind of thing happen where the Skeptobius physically attaches to the ant. So it's about as close a biological association between two animal species as you can imagine. These beetles are almost permanently latched onto the ant hosts, who really don't seem to care about it at all. And you have to wonder, how is the beetle able to do this? Because, you know, ants have this cuticular hydrocarbon code, which they use to recognize nestmates that they touch. How is this beetle able to kind of break through that and latch onto ants and with the ant tolerating what's going on? Well, This is the cuticular hydrocarbon profile of the ant. Okay, you can see all of these peaks here, alkanes and alkenes, these long chain hydrocarbons that the ant is secreting onto its body surface and act as a kind of pheromonal code for the worker ants in the colony. If we look at what the beetle is doing using high-speed video, we see they climb up on the ant's body, they bite on the first ant antennal segment, this long segment here. The beetle's got its mandibles wrapped around there to anchor itself. And look what it's doing with its legs. It's scraping against the ant's body and smearing its legs over its own body. And what it's doing is it's stealing the cuticular hydrocarbons from the ant. It's grooming the ant to physically procure cuticular hydrocarbons from the ant and smear them over its own body. And this mechanism is so effective that if you take this beetle from this ant and look at their cuticular hydrocarbon profiles, they match each other perfectly. So there's this amazing mechanism of chemical mimicry that this beetle has achieved simply by not producing any of its own cuticular hydrocarbons, but physically stealing them from the ant. And also, and it should be mentioned that the beetle can get on top of the ant in the first place to do this behavior, again, by secreting some kind of compound from an abdominal gland that appears to tranquilize the ant and enables the beetle to wander on top of it and start this grooming behavior. And if you look at the beetle's feet, you can see there are these big brushes of hairs. These are specialized setae that these beetles have that enable them to scoop up the cuticular hydrocarbons and smear them over their own bodies. Now, in this group of beetles, we have so many examples of symbiosis evolving, and we're studying just a couple of them in the lab. But the repeated evolution of the symbiosis enables you to take other approaches to study what genes and molecular mechanisms may be responsible for the evolution of this complex change in life history within this group of beetles. So one of the other things we're trying to do is genome sequence whole genomes from symbiotic beetles and their free-living relatives. And if we can do this multiple times across the tree of life of these beetles to compare the genomes of symbionts to free-living species and ask what changes commonly occur when species become symbiotic. And one of the first things we found from doing this is that just like many pathogens and parasites that are unicellular in the microbial realm, we see extreme reductions in genome sizes in some of these socially symbiotic beetles. So this is a beetle here, Pseudomymeseton, that lives in Labradus army ant colonies in Peru. This is the beetle here. This is the ant here. It appears to have the smallest genome of any insect known to date, only 98 megabases. Just to compare that to Dolotia. Dolotia has a 290 million base pair genome. This one is down to 98. So it's about a third the size of a typical rove beetle genome. So we think what may be happening here is that when these beetles become symbiotic inside ant colonies and become really specialized for that way of life, they start outsourcing bits of their biology to their host societies, so they are no longer doing it, carrying out these processes themselves. And so there's no longer any selection in the genome to maintain certain genes or signaling pathways, and this is reflected in the reduction in genome size of these beetles. So what's happening at this organismal social realm really mirrors what's happening at the microbial realm when parasites and pathogens also show this really significant gene loss as they become symbiotic and dependent on their host organisms. So what are we trying to do with this system? Well, the basic question we're asking is, how does one species of organism evolve the means to recognize and interact effectively with another species of organism? And you can see, I hope I've convinced you, that really rove beetles are unparalleled in their capacity to forge these links across species boundaries in the most remarkable ways. And so by studying these relatively simple ant-beetle relationships, which are tractable in the laboratory, we're shedding light on a really pervasive phenomenon in the natural world, which is the evolution of biological associations. And I will stop there. Thank you all for listening, and thank the members of my lab who are currently engaged in this work, and all of the agencies which have started to fund this work since I moved to Caltech in 2017. And I will stop there and take any questions. Thank you.
Thank you, Joe. So it looks like we have a few questions from the audience. The first question is, if acceptance is achieved by chemical means, what is the pressure for morphological changes?
That's a great question. Well, by and large, acceptance is achieved by chemical means, we think. But in certain special groups of ants, we think that largely the army ants here, there seems to be an additional pressure to conform to tactile nestmate recognition systems. We think that army ants are special because they form these huge collective movements across the forest floor. They're forming bridges out of their bodies. They're constantly feeling each other with their antennae. And we think that there's a kind of tactile assessment that these beetles also have to conform to integrate socially into army ant colonies. You don't see that happen inside colonies of ants that form permanent nests. They do not form these huge collective movements. There's probably less pressure for the beetles to undergo changes in shape and start resembling the ants. So it's really seen in the beetles associated with army ants. The other case where you see it in species that live inside termite colonies, and termites famously use a lot of tactile cues as well as chemical cues to chemical nestmate recognition cues. So in both those cases where we posit there's a strong use of a tactile nestmate recognition, that's when there's selection pressure on the beetles to change shape. And I should add that some of these beetles, what appears to be most important is not looking like an ant, but feeling like an ant. So for example, these Labradus army ants here, which are basically blind, these guys, you know, they emigrate at night. And they're largely subterranean. And the beetles, these guys here, they look like ants, but they don't have the same color. So there's not visually they're not amazing mimics, but they feel enough like an ant, I think, to gain social acceptance inside these nests. There's one view that these beetles mimic the ants, so they avoiding predation from vertebrates, things like, you know, amphibians or birds that may pick off organisms that the army ants flush out of the undergrowth as they emigrate. But the fact that many of these beetles don't match, they stick out like a sore thumb, even though they match the same, approximate the ant shape, suggests that the beetles are fooling the ants with their shape rather than any visually guided vertebrate predators.
Okay, our next question asks, why do the rove beetles generally seem to have an upturned abdomen? Does that mean something to the ants?
Interesting. So the rove beetle abdomen is really flexible, and when it's raised like that, that kind of means it's kind of armed and ready for chemical secretion. And so I think when the beetle is at its most alert, you know, when it's interacting with the ants, you know, its abdomen is just poised to be able to secrete these compounds from glands. So I think yeah, well, when you see that, that's the beetle kind of, you know, out of its resting state. So normally when the beetle's kind of isolated and on its own, often it's just abdomen's down. But when it's interacting with ants, the abdomen is up and it's kind of primed for chemical secretion.
Okay, our next question asks, have you analyzed the compound that the rove beetles secrete to control their hosts?
Well, that's one thing we're doing in the lab right now. We've identified some of them for the species that we're working on. The volatile compounds are things that we can pick up using gas chromatography, mass spectrometry, and they are quite easy to identify. Some of the other ones that the ants actually like feed on directly appear to be non-volatile compounds and complex blends of substances, which are proving more challenging to identify. But they are, you know, particularly interesting. So these kinds of things take time, but we are on it.
Okay, our next question asks, if there are multiple symbiotic rove beetles in an ant colony, are they cooperative with each other, ignore each other, or contest with each other?
That's a great question, and we're really interested in that kind of level of interaction between multiple invasions in the same ant colony. For the species that we study locally that live with velvety tree ant, we find 3 different lineages have infiltrated those ant colonies, and they seem to, utilize different strategies and live in different parts of the nest. So we see Platyusa, the first beetle I talked about, which has that kind of appeasement gland that uses really to kind of keep the ants at bay. That lives on the nest periphery and feeds on things like injured workers and ant larvae. And then Skeptobius is right at the other end of the extreme. This is the one that sits on the ants and grooms them, and that can be deep inside the nest. And so the chance of those beetles, I think, they don't really have that much niche overlap. They both probably use the same kind of mechanism, ancestrally to gain entry to those nests, but they've evolved, using their kind of defensive glands, which have predisposed them to become symbiotic. But then one of them, Skeptobius, has become really highly integrated. And you could argue that maybe it's much more specialized than Platyusa. Skeptobius's defensive gland is degenerated evolutionarily. It's using a specialized appeasement gland to stun the ant, climb on top of it, do this grooming behavior. It can no longer make any of its own cuticular hydrocarbons. Platyusa, at the nest periphery, still has the defensive gland, chemically mimics the ant a bit by synthesizing most of the main peaks in the cuticular hydrocarbon profile, but they're living in different kinds of niches. The question becomes particularly poignant, I think, when you see things like army ant colonies, where you get multiple ant mimics of different genera all walking kind of side by side in the same colony. And you have to wonder, in those cases, are they actually in competition with each other? Many of those beetles are quite rare inside colonies, 1 in 1,000 or 1 in every 10,000 ants or something like this. So maybe the low density of these beetles, the low frequency that you tend to find them inside nests, enables them to coexist without much kind of competitive exclusion.
It looks like this is going to be our final question for today's webinar. The question is, how much variation is observed in the appeasement chemistry among different ant species?
Oh, but among different beetle species that have the appeasement chemistry? So as far as we know, very, very little. So, but then again, nobody has done an exhaustive study of this across species. Behaviorally, most of these compounds are exerting similar effects on the ants, and our own studies where we've looked at these appeasement compounds in the laboratory, in the limited sampling of species that we've studied, we see really like very, very little variation. One thing we do find in the lab, in some of the species that we can maintain in the lab, is that after a while, culturing them in the lab, and this refers really to the free living species, and their defensive chemicals, the composition of the glandular secretions in the defensive gland can change over time depending. So you can find some of the components of the defensive secretion, things like the alkanes and esters, which act as kind of solvents for the main benzoquinones. The ratio of these things can change depending on what you feed the beetle. So there may be dietary modulation of the appeasement compounds as well. And who's to say they're not actually getting some of these compounds from the ants, or at least precursors of them from the ants themselves, because that's what many of these beetles seem to feed on.
Okay. Well, thank you, Joe, for leading a very engaging and informative seminar today. We got great feedback from the audience. I'd like to thank our audience for joining us today. Please take a moment and fill out the survey at the end of this webinar, and be sure to join us on September 17th for our webinar with career coach Sabrina Woods on building resilience to manage stress. On behalf of the Caltech Alumni Association, I hope you have a great rest of your day.